Ever wondered what goes into the process of RT-PCR? Welcome to the fascinating world of molecular biology, where we decode the secrets of life. RT-PCR, or reverse transcription polymerase chain reaction, is a laboratory technique that amplifies a single or few copies of a piece of RNA across several orders of magnitude, generating thousands to millions of copies of a particular RNA sequence. It's like making a whole army from a single soldier. What's even more interesting are the 6 key components that make this process possible. These include the nucleic acid sample, reverse transcriptase enzyme, DNA polymerase enzyme, primers, deoxynucleotide triphosphates, and PCR buffers. Each of these elements plays a vital role in ensuring the accuracy and efficiency of the RT-PCR process. But what exactly do they do? How do they contribute to this complex process? Now, let's delve deeper into each of these components. Firstly, the nucleic acid sample. In RT-PCR, RNA is the sample. Now, you might be wondering, why RNA? Well, it's mostly because the majority of the time we're dealing with mRNA. This type of RNA is the blueprint for protein synthesis in cells. By analyzing mRNA, we can get a snapshot of the genes that are active at any given time in a particular cell or tissue. Now, unlike other PCR techniques that use DNA as their sample, RT-PCR uses RNA. But there's a twist. Before we can amplify this RNA, it must first be converted into something called complementary DNA or cDNA. This conversion is an essential step in the RT-PCR process because DNA polymerase, the enzyme responsible for amplification, can only read DNA. And there you have it. That's the role of RNA, our first key player. Next, we have our enzymes, the reverse transcriptase and DNA polymerase. These are the heavy lifters in the RT-PCR process, each playing a crucial role in the conversion and replication of our RNA sample. Let's start with the reverse transcriptase enzyme. This is the enzyme that takes the lead in the first step of our RT-PCR process. It's a catalyst for the formation of complementary DNA strands from our RNA strand. This is why it's also known as an RNA-dependent DNA polymerase enzyme. In essence, it's responsible for reversing the central dogma of biology. This enzyme is the star of the show in RT-PCR, as it's the one converting our RNA sample into cDNA, which then becomes the template for amplification. Following the reverse transcriptase, we have the DNA polymerase enzyme. This enzyme is the master builder, constructing complementary DNA strands by meticulously assembling nucleotides in sequence according to the template strand. The most commonly used DNA polymerase is the taq-DNA polymerase, which is extracted from the bacterium Thermus aquaticus. This enzyme is particularly favored because it's thermally stable, meaning it continues its activity even after repeated cycles of heating and cooling. In other words, the reverse transcriptase is the architect, sketching the blueprint for our cDNA. The DNA polymerase, on the other hand, is the builder, following the blueprint to construct the complementary DNA strands. Together, they form a powerful duo, facilitating the conversion and replication of RNA into cDNAs, which can then be amplified and analyzed. It's important to note that while these enzymes are crucial, they don't work alone. They rely on an entire team of components, including primers, deoxynucleotide triphosphates, and PCR buffers, to successfully carry out the RT-PCR process. But we'll delve into these other players in the upcoming scenes. For now, let's appreciate the hard work of our reverse transcriptase and DNA polymerase enzymes. Without them, the magic of RT-PCR simply wouldn't be possible. These enzymes are the workhorses of RT-PCR. Now, on to the primers. These are the signals that guide the enzymes in RT-PCR. There are 3 different types, each with a specific role. First up, we have random primers. These are short sequences of 6 to 8 nucleotides that bind at any complementary site on the RNA. They don't discriminate based on the presence or absence of a poly-A tail. Instead, their function is to synthesize cDNA using reverse transcriptase. Next, we have the oligo-DT primers. These oligonucleotides, usually 12 to 18 nucleotides long, contain a repeating segment of deoxythymidine or DT. Their job is to bind at the polyetail of mRNA, which is a unique feature of these primers. Finally, we have sequence-specific primers. These are short sequences of nucleotides that target a specific region of interest on the sample RNA. These primers are typically used in one-step RT-PCR, where both reverse transcription and PCR happen in the same tube. So, in a nutshell, we have random primers for general cDNA synthesis, oligo-DT primers for mRNA targeting, and sequence-specific primers for precise targeting. Each of these primers plays a key role in the RT-PCR process, guiding the enzymes to the right spots and ensuring the accurate synthesis of cDNA. That's how primers guide the process. Lastly, we have the deoxynucleotide triphosphates and PCR buffers. Deoxynucleotide triphosphates, or DNTPs for short, are the building blocks of our genetic material. They are artificially synthesized nucleotides that play a crucial role in the RT-PCR process. Specifically, they are used for synthesizing complementary DNA, cDNA, and new cDNA strands during amplification. There are 4 different types of DNTPs used in this process: deoxyadenosine triphosphate, DATP, deoxyguanosine triphosphate, DGTP, deoxythymidine triphosphate, DTTP, and deoxycytidine triphosphate, DCTP. Now, let's talk about PCR buffers and other chemicals. PCR buffers are essentially solutions that provide an optimal environment for the PCR reaction to occur. They help maintain the correct pH and salt conditions that are necessary for the enzymes to function effectively. These buffers often contain potassium and magnesium salts, which are crucial for the activity and stability of the DNA polymerase enzyme. In addition to the PCR buffers, there are other chemicals that are also important in the RT-PCR process. These include chelating agents and detergent, which help to protect the DNA during the denaturation step, and an anti-foaming agent, which prevents the formation of bubbles that could interfere with the reaction. So there you have it. Deoxynucleotide triphosphates and PCR buffers, along with a few other chemicals, are the unsung heroes of the RT-PCR process. And that's the final piece of the puzzle. Let's quickly recap what we've learned today. We started with the nucleic acid sample, which in the case of RT-PCR is RNA, primarily mRNA. This RNA is converted into complementary DNA or cDNA before amplification. This conversion is facilitated by the reverse transcriptase enzyme, a vital component of the RT-PCR process. Next, we discussed the DNA polymerase enzyme, particularly the thermally stable Taq DNA polymerase, which synthesizes the complementary DNA strands. We then delved into the 3 types of primers used in RT-PCR: random primers, oligo-DT primers, and sequence-specific primers, each with their unique roles in the process. We also touched upon deoxynucleotide triphosphates, or DNTPs, which act as the building blocks for cDNA synthesis. Finally, we covered the PCR buffers and other chemicals that are essential to the process. Remember, each component has a crucial role to play in the RT-PCR process.